Alberta. This may surprise you because of how young he looks, but he and I actually did residency together. I just got older, and he, stayed the same or got younger, like Benjamin Button. I'm not sure, but he's also much smarter than me. So instead of doing intervention, he decided to take his talents down and did a fellowship in California where he learned about stroke genetics. So, been long overdue getting you up here to give this talk, and I'm really pleased that you're here. Welcome.
Yeah. Yeah, thank you very much for the invitation. Such a great conference. Thanks for having me participate. Going a bit backwards from what Ashfaq was presenting, but my objectives are discuss about omics and stroke, what is maybe some stuff that's coming. And so what is omics? It's the evaluation of not just the DNA, but also the epigenome, how the genes are expressed, the influence on the proteins and the metabolome. How does it integrate together to influence whether you have disease or not? And so what's in the world of stroke for this and what might be coming is what I'll be talking about. So why omics? I think it provides a good insight into disease that may not be available from single molecule evaluation. Stroke is quite a heterogeneous disease, and by taking a broad approach, it can allow a screening of a bunch of molecules and proteins and metabolites that might be influencing disease risk. Most of the work in stroke in terms of at least biomarkers has been done at the protein level, and a lot of the proteins have lacked sufficient sensitivity or specificity to be clinically useful in stroke. And so the hope is maybe by taking a broader approach, screening many molecules, more sensitive panels could be identified that might be helpful clinically. In terms of a system approach, I think this world initially started off at the genome level, where you'd identify SNPs or genetic variants that might be associated with a disease. And sometimes these were related to disease, but there was a realization that this doesn't always translate to a clinically relevant phenotype. And so looking deeper, what is the influence of the epigenome, the transcriptome, proteome, metabolome, and even microbiome on how does this relate to disease? And I'm going to there's quite a lot of work on this. I won't be summarizing most of it. I only have 3 topics. One, genome first, or first I'll talk phenotype first. What are 3 things in stroke where omics have had some demonstrated use? I think this is mostly where it's useful clinically. A clinical phenotype, if you have a clinical phenotype that's of interest, like etiology of stroke, this phenotype first approach, what are the omics that are different in that, can provide some insight. The other approach has been a genotype. So say you have a SNP that is known to be associated with a disease, what is the changes in the omic profile? So in terms of phenotype first, I'll talk about stroke risk, hemorrhagic transformation, and stroke etiology. And in terms of genotype first, I'll talk about 3 SNPs, HDAC9, CYP2C19, and the Notch3 mutation. So phenotype first, stroke risk, we've already heard about the polygenetic risk score, but also metabolites. I think we frequently use glucose or lipids. Already some metabolites are used to assess stroke risk, but there may be some more stuff coming in the metabolite world. Metabolites are typically screened by a mass spec profile. To date, there's been maybe about 5-10 studies profiling stroke patients, comparing stroke to control patients, and identifying metabolites that might be different in the plasma. And there have been a lot of metabolites identified. The problem has been it's been inconsistent between studies. Metabolites are quite sensitive to diet and other metabolic changes. And so I think the challenge is getting large enough cohorts to identify a consistent signal what's going on in the metabolites. But I think there's emerging data that age is strongly linked to changes in metabolism. There's marked changes in our metabolites at age 40 and age 60. And given age has such a strong influence on stroke risk, there likely are some of these metabolites that do influence stroke risk. So I think probably in the next 5-10 years, increasing data on metabolites that could be targeted to assess and potentially treat stroke may be coming. The polygenetic risk score, I think we had also heard about. This was a nice study. I think it also echoes Ashfaq's point that in the clinical factors are still super important, and it's if you have to pick one, pick the clinical factors, especially hypertension. This was a study comparing the risk of someone's stroke. In red is showing some genetic risk scores. It's a 90 SNP score. 90 individual measures of SNP associated with stroke. And even with this 90 SNP score, it's not even close to what is assessed by hypertension or some of our risk factors. The interesting part, I like it that clinical risk factors actually contribute a lot. So we're doing the right stuff by targeting hypertension, diabetes, and so this global health worldwide, targeting that before genetics, I think is important. But there is some additive. So in the lower two red bars, that's the combined, especially the lower one, combined genetic does add a little bit to these clinical risks factors in terms of causing strokes. So there may be added benefit to assessing genetics to it and understanding what future targets might exist in terms of reducing stroke risk. Another phenotype in stroke may be hemorrhagic transformation. Often when we treat with TPA, we're worried what is the risk of hemorrhagic transformation. Typically, we'd say 6 to 7% risk of hemorrhagic transformation. In this study, we looked at blood cell gene expression in patients with the blood drawn just before TPA was given. And we could, by just looking at blood transcriptome levels, put them into two categories. One group shown in the red had a 80% chance of having a hemorrhagic transformation at 24 hours, whereas the ones in blue were almost zero. And so there may be strategies in people with milder stroke to guide whether, if it's a milder stroke, maybe the risk of hemorrhage is too high, or there may be alternative targets such as targeting the immune system or neutrophils to reduce that risk and make TPA safer. The last phenotype I wanted to talk about was etiology of stroke. So I think we had heard about ESUS patients earlier today. It's a challenge to identify what is the cause of stroke, super important to preventing stroke. And so could additional markers such as omic markers be valuable to identify people with cardioembolic stroke or large vessel stroke? This was an initial study where we'd screened blood of patients with clear arterial and large vessel stroke and lacunar stroke and found that there are transcriptomic differences in these patients. This relates to aspects of blood clot formation in the cardioembolic stroke, as well as it seems to be auto-advancing, but immune aspects and related to plaque rupture and plaque formation in the cardioembolic or in the large vessel stroke. And so potentially this might add to cardiac measures or vascular measures to help refine what is the etiology of stroke. Let's skip that. And I guess the last phenotype we're starting to look at is in terms of clot properties. Initially, we were thinking clot is clot, and there's not that much differences, but increasingly we're realizing there's quite a lot of heterogeneity in a person's clot when it gets taken out by endovascular therapy, and this can influence treatment response, recanalization success, and so potentially by better understanding clot properties using omics, you could gain better insight. Could we target properties of the clot beyond thrombolysis with fibrin? Could other properties of the clot be targeted? And some of the insight to that is being revealed by omics. So we're hopeful that in the next 5-10 years, we might have ancillary stuff to target in the clots beyond just fibrin lysis. So in terms of genotype first, this will be a bit faster. There's 3 SNPs I wanted to talk about. So the first one is HDAC9. This is a SNP that's been strongly associated with large vessel atherosclerosis, and patients who have this SNP tend to have a higher risk for large vessel stroke. But it's a bit unclear, what does this HDAC SNP do in patients? And so we had screened patients who have this SNP, large vessel stroke with the SNP or without, and there are transcriptomic differences in the blood of these patients. There seems to be potentially a change in a number of factors, including T cell response and immune activation, that may influence potentially propensity for plaque formation or plaque rupture. And so this gives insight to a patient's risk of atherosclerosis. It doesn't really influence treatment at the moment, but it can provide sort of more refining of someone's etiology of stroke. This has already been discussed. The CYP2C19 variant has been known to influence the metabolism of Plavix to get it to the active metabolite. Initially, very promising. I think there was hope that you could guide therapy by testing this SNP, but I think when it's been checked, this didn't necessarily translate into patients being stratified into higher risk groups or not, or benefit of Plavix or not. And so some people have gone back and looked at in people who have the CYP2, this CYP variant, are there other factors downstream in the epigenome and the metabolome or microbiome that might be, it can also influence how does Plavix get metabolized. And so it may be a bit more complicated than checking one SNP. You can have the SNP but still be able to metabolize Plavix in alternative methods, or you might not have the SNP, but you still have an impairment in metabolizing Plavix. And so it might need a bit more comprehensive screen to say, is someone a good response? Does someone have a good response to an antiplatelet such as Plavix or not? And finally, Notch3 mutation, well known to be the important on CADASIL. What's been interesting is you can have patients with the exact same Notch3 mutation, but different phenotype or severity of disease. And what explains that? And so there may be downstream aspects in a person's either different genetic variants that add to the Notch3 mutation to add to that variation, but also differences in the transcript, how they're transcribing, epigenetic regulating. People have also taken people with a Notch gene, people with CADASIL, and looked at their brain and tissue and looked at omics changes, and that's revealed some interesting stuff in terms of the blood vasculature, the brain tissue, and it's providing insight to the disease. So it's a way omics can provide insight to stroke using genetic stuff. And so I guess this is kind of summarizing back, probably clinically the best is taking a phenotype first and then can screen through things at a omics level, either to develop biomarkers or gain insight to your disease. And then genotype, if a genetic variant is identified, you can gain insight into that. So I think it's an emerging area of omics. I don't think there's really yet a clinical application of it, but I think the field is growing strong, and I think increasingly genetic variants that are identified are going to have increased insight to what they're doing, how they contribute to stroke. And I think that even though there's this sort of two levels, phenotype first and genotype first, I think the next approach is how do you integrate this multi-omic approach? And so I think there's a lot of effort going on to how do you integrate the multi-omic or using probably AI to assess what's going on in our patients and how that all integrates together. So I imagine that's coming in the next 5-10 years. So thank you very much.
Great, thank you. And now we can move on to our panel. So